Role of phytohormones to improve seed germination. Here, seed germination and role of plant growth regulators, also we call it as a phytohormone. So different types of phytohormones, they are produced in the plant body. They regulate the growth of the plant, and they are important in the life cycle of plants. Also in process like seed dormancy and also in the process of seed germination, phytohormones have their crucial role. Means we can say that dormancy as well as germination, both these crucial phases in the plant life cycle, they are controlled by phytohormones. The process of seed dormancy and germination, actually they are antagonistic to each other. Both the processes, they are controlled by and regulated by diverse endogenous hormones and also environmental factors. Abscisic acid and gibberellic acids are the major determinants of seed dormancy and germination. Abscisic acid and gibberellins, both antagonistically regulate seed dormancy and germination. Gibberellic acid is a phytochrome involved in seed germination and responsible for breaking seed dormancy. It stimulates germination by reversing the effect of abscisic acid. Gibberellic acid stimulate the biosynthesis of hydrolytic enzymes like amylase, peptidase, proteases, and lipase. These enzymes break down complex reserve food, for example, carbohydrate, protein, and also fat, as well as oil, and produce simple consumable substances like sugars, amino acids, fatty acids, and triglycerides. These substances are then used by developing embryo during germination process, leading to enhancement of germination. Other plant hormones like auxin, ethylene, brassinosteroids, jasminic acid, salicylic acid, cytokinins, strigolactones are also involved in seed germination activity. Out of these, especially ethylene is responsible for breaking seed dormancy, and this phytohormone ethylene promotes seed germination by counteracting effect of abscisic acid. Another phytohormone, brassinosteroid, a new group of phytohormone can overcome the inhibitory effect of abscisic acid and stimulate germination of the seed. Salicylic acid also regulates seed germination. It promotes germination under high salinity conditions. Cytokinins is another group of phytohormones responsible for seed germination. It promotes seed germination by antagonizing effect of ABA or abscisic acid. Jasmonic acid is one more phytohormone which suppresses the genes responsible for abscisic acid biosynthesis, and therefore ABA will not be synthesized, and in absence of abscisic acid, seed germination process gets accelerated. So this is the survey of different phytohormones involved in the seed germination process. Figure index. Seed start germination. When it happens, it is interesting to note or record various parameters like percentage of seed germination, what is the root and shoot length, root and shoot ratio, mobilization efficiency, germination index, and vigor index. The quality of seed and germination ability of the seed is tested by such parameters. Such parameters give an idea about use of seeds for sowing and raising new crop in agriculture. Seed vigor is an important quality parameter which needs to be assessed to supplement germination and viability tests to get insight into the performance of seeds in the field or even in storage. One can define seed vigor as the sum total of those properties of the seed which determine the level of activity and performance of the seed or seed lot during germination and seedling emergence. Like figure index, other parameters like seed index and germination index are equally important. Speed of germination and time required for germination are also key parameters in seed germination. What is the importance of vigor index? That we will see. So in case of germinating seed, not only vigor index is important, but time, rate, homogeneity, and synchronization of germination are crucial issues. simply because the dynamics of germination process are indicative of ecology, physiology, phenology, growth, productivity, and development of particular plant species. Using these indices, ecology of plant population could be better predicted. Vigor index and other parameters are important to know successful establishment and survival of the plant species in nature or in particular habitat over a time period. How vigor index is calculated, that we will see now. So vigor index is equal to germination percentage into seedling dry weight. It can be calculated as germination percent into average seedling length even, or vigor index is equal to germination percent into shoot length.